As you have seen, the reversal of DNA damage by using single enzymes, but there are many mechanisms, many types of DNA damages cannot be repaired by a single enzyme that catalyzes direct damage reversal. Instead, repair requires the participation of several different enzymes, each performing a specific task in a multi-step pathway. Damage to DNA bases caused by deamination, oxidation, and alkylation is mainly repaired by one such multi-step pathway, which is called base excision repair or BER. BER pathway is same in all the organisms. Enzymes involved in the base excision repair pathway also participate in the repair of single-stranded break in DNA. So these enzymes which are involved in BER pathway, they can also repair single-stranded breaks. Base excision repair derived its name from the first step in the pathway, that is N-glycosyl bond cleavage. So in this pathway, the first step is the cleavage of N-glycosyl bond, and by the cleavage of this bond, the base is removed, nitrogenous base is removed or excised from the DNA helix. So that's why it is called base excision pathway. This cleavage excises the damaged or inappropriate base from the DNA to form an abasic site, that is a site without any base. Because no single enzyme can distinguish the four bases present in DNA from a wide variety of altered bases. There are many types of altered bases and four types of normal bases. So no single enzyme can make distinction between all these nitrogenous bases. So cells must use many different enzymes to perform this function. Some N-glycosylases are monofunctional enzymes with only the ability to excise a damaged base. They have only one function, that is to recognize the damaged base and to excise that base. Such enzymes are called DNA glycosylases. While other N-glycosylases have an apelase activity that cleaves the bond between sugar and the phosphate 3' to the damaged site. So these glycosylases can introduce a cut. This enzyme is designated as DNA glycosylase lyase. So here you can see this is the reaction catalyzed by DNA glycosylase, which is a monofunctional enzyme. So this is a DNA with damaged base. This is here a damaged base is present, and this damaged base is linked with the sugar through this glycosyl bond. So in this reaction, this damaged base is directly removed, and here an abasic site is present. So because the damaged base which was present here, that has been removed. So this is an abasic DNA. So in this way DNA glycosylase works. So this is the example of reaction catalyzed by DNA glycosylase lyase activity. So in this case again, this is damaged base, and this base is directly removed after the excision, after the cleavage of this glycosyl bond, this bond, and this DNA is produced with a basic site. There is no base. Okay. So the second step is the cleavage of this bond, this 3' linkage between phosphate group and the sugar. So this bond is cleaved. So in this way, this DNA glycosylase, it performs two types of functions, that is N-glycosyl cleavage and this ester bond cleavage. Both types of enzymes detect the damaged base, flip it out of the DNA helix into an active site pocket of the enzyme, and then cleave the N-glycosyl bond. Although some glycosylases excise a specific base, most of these enzymes uh, have a somewhat broader specificity. They can excise number of bases. The E. coli enzyme uracil N-glycosylase, that is UNG, is specific, that is specific only for uracil, while another E. coli N-glycosylase, which is 3-methyladenine DNA glycosylase, which is also called ALK-A. It acts on 3 or 7 methylpurines, 3 or 7 ethylpurine, ethanoadenine, and O2 methylpyrimidine. So it has very broader specificity. It can act on a number of altered bases. So a null mutation in a gene that codes for a DNA glycosylase or DNA glycosylase lyase is not lethal, which probably reflects overlapping functional abilities among the glycosylases.